finally figure out that you have Lyme disease. And who knows how long that took you, because it can be a journey. So you go to the doctor and they say, no worries, I'm gonna give you a prescription for 28 days of doxycycline and you're gonna be fine. But is that really true? Does the antibiotic get rid of the infection? What happens after treatment? How does your immune system respond? These are some of the core questions that the person I'm speaking with next on our Lyme Warrior research interview series has been studying for years. She's been looking at groundbreaking science, trying to uncover these core issues and figure out the best methods of treatments and the best combinations of antibiotics to get rid of Lyme. So you're gonna wanna hear what she has to say. Hey everyone, my name is Nicole Bell and welcome back to the Lyme Warrior interview research series. I am super excited today to talk to Dr. Monika Embers. She is the Associate Professor of Microbiology and Immunology and the Director of Vector-Borne Disease Research at Tulane University and the National Primate Research Center. Her work and her research is at the center of all sorts of key questions that relate to the Lyme epidemic. She looks at antibiotic resistance of the bacteria. She looks at new treatment methodologies. She looks at the immune system's response to the bacteria. All of the core things that have been hotly debated, where there's tons of misinformation out there, she has been doing fundamental and groundbreaking science to try and figure out the answers and to figure out the right sorts of treatment for the Lyme patients. And so I'm really excited to talk to her today, and I think you're going to enjoy our conversation. Okay, so thank you, Dr. Embers, for joining us today. I have to say, it's a pleasure to meet you.
Well, thank you so much. Likewise.
So I have to admit, I've wanted to meet you for a really long time because I'm a big fan of your work. I was a caregiver for my husband who was diagnosed with neurological Lyme disease, Bartonella, and a whole host of other things. And I'm a scientist and engineer, so I just dove in, right, and tried to learn as much as I could about this disease and how the treatment, went and what the right approaches are. And I have to say, I was like immediately frustrated because all of the evidence was all over the place. You would read one study that was saying something else, and then something else was over here, and the experts couldn't agree. And I was really frustrated. And then I came across your research, which I really just enjoyed it because you're really focusing on some of the core issues that are at the heart of a lot of these really critical debates. And so, first of all, I just wanted to say thank you, because I think that this, the areas of study that you're going after are really critical for Lyme patients. And then secondly, I wanted to focus our discussion today on kind of debunking some of that misinformation and really focusing on the science behind some of these issues.
Sounds great. Let's go.
Good. All right. So the first thing that I struggle with, and I think probably all Lyme patients struggle with, is the standard of treatment, right? So the ISTA recommends 28 days of doxycycline. This is supported by the CDC. These are organizations that are very well respected and that we should trust when we come to recommendations. But yet there's all this information and other experts out there and lots of patient testimonials saying that this isn't effective and that people are persisting in their illness. And so you've done a ton of research specifically on how Borrelia responds to doxycycline. So can you talk about how doxycycline works in this bacteria and how it responds?
Certainly. This is something that we've been focused on for a long time, and that's because doxycycline is by far the most commonly prescribed antibiotic for Lyme disease or Lyme borreliosis. And doxycycline is a drug that is considered to be microbiostatic, meaning that it doesn't actually kill the bacteria, but it acts on actively growing cells. So what we have found is that the bacteria, the Borrelia burgdorferi bacteria, can become tolerant to that antibiotic because they enter this slow growing dormant phase. And it's not surprising that they should have a dormant phase or a phenotype because they survive for months and months inside ticks without nutrients. And so it's part of their life cycle to enter dormancy. And the reason that we the reason that we started studying doxycycline is because of the large number of patients who continue to have symptoms after the standard recommended treatment. And, I like to say I didn't make up the problem. post-treatment Lyme disease. I just am trying to figure out how to fix it, right? And so the easiest hypothesis to test is that of a persistent infection. And so several years ago, we went about doing this and realized how difficult it was, because it's so hard to see the infection, to recover the spirochetes, to reculture them. And there have been a lot of different studies done in mice. And I think Stephen Barthold and Amir Hadzik at UC Davis sort of set the stage with their studies in mice, showing that the spirochetes could persist after standard recommended antibiotic treatment. And then we set out to do this in non-human primates because they so accurately reflect human infection. And we had a hard time finding them. So we used xenodiagnosis, which is where you place uninfected ticks on the animals that have been treated. And those ticks are able to take up the spirochetes that are otherwise hard to find. And then we've also looked inside tissues. And I can tell you that it takes a lot of patience and a lot of work to find very few spirochetes in animals that have either been treated or not. They're just very few, but they certainly cause a lot of problems. And so these are the two ways that we've essentially shown that the standard of care, especially doxycycline, when given after the infection has disseminated, does not cure the infection.
Okay. And in your research, as you said, you focus a lot on primates, but you've also been involved in some human case studies, and you've seen in both of those that the bacteria can persist even after extensive cycles of doxycycline. So maybe talk through some of the more notable findings there and how you were able to demonstrate that it persists.
Mm-hmm. Yeah, so last year we published a case report of a woman who had been diagnosed with Lyme disease with a classic bullseye rash, treated with doxycycline, and showed initial improvement. And then over time, she declined and was given a more extensive and aggressive treatment, including intravenous antibiotics. And she improved to about 60%. And then over time developed signs of Lewy body dementia and other neurological complications. Eventually she was unable to eat and passed away. And we received her brain and spinal cord from Columbia University. This is a collaboration with Dr. Brian Fallon and Dr. Andrew Dwork. And we identified the Borrelia in her brain and spinal cord. And what's interesting is we continue to look at these tissues and we continue to find spirochetes. So I don't think it's a direct link between a Borrelia burgdorferi infection and the development of dementia, but I do think that patients who have Lyme disease can progress to dementia, and that antibiotic treatments don't always work. It's clear to me that it's going to take something, once the infection has disseminated, it's going to take something more strong to clear the infection. So that's where we're going now with our research.
Okay. Well, in terms of, and I think we'll come back to that, but in terms of the actual bacteria, right? So surviving antibiotics is one thing, but surviving your immune system is another. And we have a very complex immune system with lots of defense mechanisms, but yet the bacteria somehow still is able to evade that. And that's another huge area of your research. And so, maybe just talk a little bit more about why the bacteria are so stealthy, how they hide and evade detection, and why a patient who's been infected to the point where they're, severely ill can still test negative on the standard test looking for antibodies.
Yeah, I think that's another facet of the zoonotic nature of this pathogen, because they have evolved, these spirochete bacteria have evolved in nature to survive in the reservoir host with limited detection. And so in their natural cycle, they're living happily inside the mouse host or rodent a variety of rodents without inducing the kind of immune response that would kill them or eliminate them, so the ticks can come and pick them up and transmit them through their cycle. And so I think that most of the same things that they do to evade host immunity in mice, they also do in us. We're incidental hosts in that cycle. And so they have become stealth pathogens. And this is really what interested me initially in Lyme disease and Borrelia burgdorferi is their keen ability to evade all of the host defenses. And there's some pretty solid evidence that they can also suppress antibody responses, not just evade them. So, and this can affect B cells, which produce antibodies. So it's not unlikely that a patient could be infected with Borrelia burgdorferi and their B cell responses are affected. So, and that's what we use for diagnostics. So it ends up being a difficult test. And I think, we have a good bit of research, and there are some published studies as well, which indicate that the patients who develop good antibody responses to begin with are the ones who go on to recover with antibiotic treatment. And it makes sense if you're using doxycycline that you need that antibody response or that host immune response to help. Mm-hmm. But patients who develop poor responses to begin with are the ones who tend to not recover and develop post-treatment Lyme disease.
Okay. And have you found any linkages between kind of, the why some patients are having the stronger response and others are not, or is that still ongoing?
It's still ongoing. It's something that we've seen in the non-human primates, which means that it's host dependent, because we can infect 10 different rhesus macaques with the same strain of Borrelia burgdorferi and get 10 different outcomes, just like the human population. So there's variation in the bacteria, but there's also variation in the host. And I think it's highly host dependent.
Well, and I love that you say that, because that's exactly one of the other core struggles that I had is really understanding how one bacteria can lead to so many different responses. And if you read some research and the conflict that exists, it's like, well, Lyme leads to rash and a fever, right? And then you talk to other experts and they're like, well, actually it can be the whole host, the root of a lot of chronic illnesses, including neurodegeneration, arthritis, carditis, like all sorts of things, which seems, kind of crazy if you're just getting into the space. Like, how can one bacteria lead to so many different phenotypes? But you've seen that, as you just said, the same strain in your monkeys can lead to very different reactions. So can you just unpack that a little bit more and talk about the range of symptoms that you're seeing in your research and why you think that that's the case?
Yeah, so we don't really know what dictates where the spirochetes go in the host or how they affect the tissues, but we know that certain organ systems are affected more frequently than others, including, the heart and the joints and the central nervous system. But like I said, we can infect 10 different rhesus macaques, and one animal will have carditis, one animal will have joints affected. We see peripheral nerves affected. And what we commonly see is mild to moderate inflammation. We don't often see gross pathology in these animals. And I think, this really speaks to the inability to accurately diagnose and see Lyme disease in patients, because you can imagine if your peripheral nerves have mild to moderate inflammation, you have a few spirochetes lingering that are just causing havoc, that you're going to get those, sort of subjective symptoms. You're going to get, have musculoskeletal pain. You're going to have brain fog if you have a few in your brain, and some mild to moderate inflammation. So it kind of makes sense to me in that even small amounts of spirochetes and inflammation can cause a tremendous amount of morbidity in patients. And then, throw in throw in on top of that the potential for co-infections, right? So, when ticks feed, they regurgitate everything in their midgut, and oftentimes it's not just Borrelia burgdorferi. So those co-infections can complicate the disease presentation as well. And we have a study that we're about to begin looking at Borrelia burgdorferi and Anaplasma phagocytophilum in non-human primates to see how both the immune response is affected and the disease presentation, because that could also affect the diagnostic tests if the antibody response is affected. So there are many factors to consider.
And so specifically in different combinations with different co-infections, are you looking into, anaplasma you mentioned? I know you've done some work in Bartonella. Like what are the studies and research that you're undergoing there?
Yeah, so we've begun a program on Bartonella research as well. And there exists a problem because a really clearly defined animal model is not available for Bartonella research. And so that's something that we've been working on developing so that we can test therapeutics for Bartonellosis. And, it's kind of controversial whether or not it's transmitted by ticks, but there are a lot of patients who appear to be presenting with Bartonella and Lyme disease. And I think that the Lyme disease probably is causing an immune suppressive phenotype in the host so that the Bartonella can become more apparent, because in healthy individuals, it typically is self-resolving. And it was originally characterized during the HIV epidemic. So immunocompromised patients were becoming infected with Bartonella and having complications. So I think that the mix of Borrelia and Bartonella is very dangerous. And this is something that we're also exploring in the lab.
Okay, great. Well, and I think the other core struggle that I had, that's near and dear to my heart is the linkage between Lyme and dementia, right? I think there's, my husband was diagnosed with early onset Alzheimer's disease, even though he had no genetic predisposition or comorbidities for that diagnosis. And then later I found out that he had Lyme and Bartonella along with a host of other conditions. And so, but if you go to talk to a mainstream neurologist about this, they will literally laugh you out of their office. And so again, the conflicting messages. And so, you've studied neuroborreliosis and Bartonella, can you talk a little bit about how these organisms infect the brain and then how they could lead to neurodegeneration and potentially dementia-like symptoms?
Certainly, these are both pathogens that you don't want to have in your brain. The microbial cause or etiology of Alzheimer's has been something that's been a bit contentious over time, but it's starting to gain more traction. And it's not just Borrelia. HSV was one of the first pathogens implicated in the development. And what I really think is happening is that the microbe induces a cascade of events, which leads to the neuronal damage. So beta amyloid production is actually a defense mechanism. So, we've used these amyloid busting drugs and they are not working because they're not getting at the source. And the source is probably the infection and the inflammation from the infection. So I think how Bartonellosis, if it gets, neurological Bartonellosis or neurolyme, how they present has a lot to do with where they end up in the central nervous system. And so, there are mental health disorders that have been associated with Lyme disease. There's dementia, there's just generalized brain fog, there's motor neuron, motor skills affected based on where it ends up in the brain. And I think there's some pretty good evidence that a microbial infection leads to inflammation, and that inflammation can produce a biochemical cascade in the brain, which leads to the production of neurotoxic chemicals. And so finding a way to stop that is also something that is of interest to our lab. And we have some ideas about that as well. So stay tuned.
Well, and I think that's a good segue into my next question, is because so far it's been a little doom and gloom, right? You know, Borrelia evades our immune system. It evades, it persists after doxycycline, and it can infect your brain. It's like all sorts of scary stuff. So, but I know that you also focused a lot of your research on kind of the solutions and looking at new therapies and testing the effectiveness of that. And so, and I want to kind of provide that hope as you know, what's in the future, and why are you feeling good about where we're going with the treatment of this disease?
Sure. So we envision Borrelia infection something like tuberculosis. And when someone is afflicted with tuberculosis, we don't treat them with a single drug. We treat them with multiple drugs that all have different mechanisms of action that end up killing the persisters. And so we're taking the same strategy into treating Lyme disease and Bartonella, actually, wherein we use double and triple combinations of antibiotics to see if we can eradicate the infection. So we start with, testing them in the test tube and then testing them in mice. And I have to thank the Stephen and Alexander Cohen Foundation and the Bay Area Lyme Foundation for funding this research, because I think it's, this is the way that we can provide critical treatment that's needed to clear the infection. I like to say that we need to arm the clinicians with the tools they need to cure the infection. Instead of having all this controversy, maybe we can prove that one treatment works better than another. And in our hands, no single drug has really been able to eradicate the infection. So we strongly believe that this combination therapy will be much more effective. We also have a collaboration with Duke University, Tim Haysted, and NC State at Bright Schwert to look at small molecule inhibitors of Borrelia burgdorferi and Bartonella. And, they do the drug discovery and I do the animal testing. And we have mechanisms in place to determine if the infection has been cleared or not, which is really the challenging part.
Can you talk about that a little bit more, just to kind of understand, because that's a key issue for a lot of patients?
It is, and there's a lot of controversy over the bacteria not being culturable, right? After antibiotic treatment, a lot of clinicians will argue that even though the bacteria are there, you can't culture them, and so they're not alive. We've done a number of tests to indicate that they are still alive, and we've also been working on a culture media that is much more capable of regrowing the bacteria after antibiotic treatment. So those are two aspects, plus the xenodiagnosis, which in our hands has been the most sensitive method to look for persistent infection. With regard to Bartonella, we performed a study where we cultured the bacteria outside of host cells and cultured the bacteria inside host cells. And we know that Bartonella can invade, they replicate in red blood cells and can invade a variety of monocytic cell types. And so when we tested antibiotics against the extracellular bacteria, they killed them very nicely. But when we tested the antibiotics against the intracellular bacteria, they didn't work at all. So then we tested combinations and found that we got much better killing with combination antibiotic therapy. So now we're hoping to do more of that with Bartonella as well.
Oh, that's great. So I think the goal is to get to kind of recommendations for clinicians about the most effective way to treat this. And I guess just going back to the different phenotypes, have you seen with when it infects different types of tissue, whether it's neurological versus carditis versus, in your joints, have you been able to find different types of therapies that are effective for those different phenotypes, or is it all kind of lumped in one bucket?
That's a really good question, Nicole. It's something that's a little bit difficult to study because once you take them out of the tissue, are they the same phenotype? But I think, it brings about the importance of knowing that you have good penetration of your antibiotic, especially across the blood-brain barrier. So these are important considerations that we're also thinking about when defining a proper tri or dual antibiotic treatment.
Great. Well, as I said at the beginning, I think a lot of these issues are just really core to the Lyme disease community and the proper way to get well. And so I really applaud you and your research group for all that you've done to kind of really dive in on some of these scientific challenges and the core issues that are surrounding this illness. So thank you so much for your work, and thank you so much for talking with us today.
Thank you, Nicole. It's been a pleasure. And I wouldn't be doing all the things that I do without generous support from foundations, the two that I mentioned before, Global Lyme Alliance, Focus on Lyme. I've had a lot of great support, and we look forward to doing better things.
Great. Well, thank you so much.